I'm Serena. You may have seen probably other webinars done on my side. I'm the Senior Informatics Solution Lead at FlowJo, and I've been working with FlowJo for 6 years. I can't say I know it all, but I know several things about FlowJo, so feel free to ask me anything you have in mind in the Q&A box. And don't be shy, absolutely. This session is for you. It's a live Q&A, so feel free to ask anything. Here on the screen, you can also find my email address, serena.di.cecilia@bd.com. I know it's quite long, and you have to remember my name and last name and remember to add a dot, because otherwise I don't get your emails. But please feel free to send me an email in case you need any help, or you do have any question or any problem, or you need to troubleshoot FlowJo somehow. I'll be happy to to support you. I'm based in Italy, so here it's 10:04, so I believe it's time to start. If anyone is based somewhere else, just type your country in the chat so we can make this a little bit more interactive. Okay, so on with the questions. If you have any burning question, please type it in the chat. Meanwhile, I can maybe start with some of the questions that I received by email. And so there is a really frequent question that it's also one of the questions that arrived, that is, can I rename an FCS file? Now, this is a really frequent question, and the short answer to this is, if you want to rename the actual FCS file within FlowJo, you can't. But there are several ways, several things you can do. So the first thing you may want to do is, if you realize that the name of your file is not what you want to see, one thing to do that makes you sure that you are renaming the file itself, it's going back to the instrument and renaming the file and then re-exporting it. The reason why it is not possible to literally rename the file in FlowJo, it's for security reasons. So we want to protect you, we want to protect your data, so you are the only one that can actually go back to the instrument and rename the file and re-export it. But in FlowJo, you can do several things to, let's say, try to work around this problem or this thing. Okay, let me just close this. Oops. And I'm going into my FlowJo. And if I add a sample now, any kind of sample, what I will see is the name of the sample that comes from the instrument. So basically, the way I named the sample at the instrument. Sometimes I get this question that says, I don't see the name of the file the way I labeled it at the instrument. I see it with like a super short file name, something that I really don't understand, and it's not the way I named them. So in your preferences, if you just click on the heart icon on the top right corner of FlowJo, you can actually go into Workspace and choose to use the short file name. The short file name is actually the way you named the file at the instrument. And you may realize that if you see a different name right here, it's because you're using the FCS default name, which is basically what was assigned from the instrument itself. Let me save this. So in case you have this FCS default sample name keyword assigned to it in your preferences, then you will not see what you name your file at the instrument. Otherwise, you can just use the short file name. In case you want to use use a different way, okay? Let's say you're not going to rename the actual file, but you can change the visualization of the file name within the workspace by using a keyword combination, for example. You can use different keywords. You assign first the keyword to the file, and then you can use that keyword to display on your workspace. But otherwise, it's really not the case of completely renaming a file, an original file within FlowJo. My advice in this case is always to use the short file name. And just we are here, I want to highlight, in case you have never seen that you can enable the auto saving of your workspace in this area right here. So when you enable the auto saving, FlowJo will save automatically your workspace every interval of minutes that you will decide. So I have it set to 5 minutes, but you can set it to 3 minutes, 7 minutes, 10 minutes, whatever you prefer. And I strongly recommend you to enable this auto saving, because what can happen if you do not manually save your workspace, and let's say you run out of battery, your computer runs out of battery or crashes somehow, you lose what you have done. So my advice, absolutely enable the auto saving, so you will be free to go. You will get the file saved, the workspace saved every interval of minutes that you decide, and you can go. Okay, so let's see if we do have something else. No questions popping up yet. Okay, so I will move on to the next question that I got from the mailbox, which is, why does my template not apply to new samples even though it's the same panel and experiment? How can I fix or prevent this from happening? Well, first of all, let's say, what is a template? A template is a workspace that you can use that has defined gates and defined layout setting, and you can use it to analyze new samples every time. So it's like, let's say, the templates that you have, I don't know, in PowerPoint or Excel or Word and so on. So it's something fixed that you can use to analyze new files every day. Now, in order to have a template to work, you need to have a gating strategy. So let's say that I will start my analysis today and I want to create a template, okay? So the first thing that I want to do is create a group. So by default, FlowJo will always show you two different groups, all samples group and compensation group. The all samples group has the function to keep all your samples together. The compensation group will have samples included in it if there are compensation controls, okay? So now, whenever we start an analysis in FlowJo, not only when we want to make a template, we should always create a group that mirror our experiment. So let's say we do have 3 different set of samples that can be full stain samples, control samples, and compensation samples. Then we will have 3 different groups. So first thing, I'm going to create my group. This will be my, let's call it full stain. Okay. And whenever you create a group, you can assign a color, you can assign style, a role. This can be my test role. And you will see that you have the option to make this group live or synchronized. So what is the difference between live and synchronized group? The live group will be automatically including all the samples in the group that have specific features, for example, that are defined here in the keyword tab right here. So for example, I can tell FlowJo to create a group and to make this group live in a way that every time that there is a sample or I load the sample into my workspace, each sample that has, for example, a keyword associated to it that can be a file name, okay, just by selecting the file name that contains a word, like in this case, I'm working with live donor, I can use LD for that. So the live option will allow the workspace and will allow the group to automatically host all the samples that in their name will contain the word LD. And this live option here is really important when you create a template. Okay, so in case you want to create a template, please have the live option on. The synchronized option is slightly different. It's related to the gates that you perform. So whenever you have a group, you can create a group-owned gating strategy, and the synchronized group will allow to each gate to be automatically adjusted on each sample. So I'm going to show it in a second. So if I now create the group and close, you see that automatically in this group, the sample that includes in the name LD has been loaded. And this is what you want to have when you create a template. Okay, so naming a file is extremely important. Name it correctly and use names that can actually help you in your analysis. Not only knowing what is what, but also to streamline your analysis in case you're creating groups, you're creating keywords, you're creating templates, and so on. So back to the template. If I start now to create a gating strategy, okay, let's say these are my lymphocytes, and then within my lymphocytes, I want just create a gating strategy that goes like with 2 or 3 gates, okay? And then I have that. So I'm gonna create like just a quick gating strategy for that. So at least we have few gates to play with. Okay. Now I have my gating strategy and I want to create a template which includes this gating strategy. So what I have to do in order to make the template work is to apply the gating strategy that I want to use in my next samples to the group. So I can select the full gating strategy just by selecting the first gate, holding down the shift key and selecting the last gate. And then I can either drag and drop this gating strategy into the group, or I can use a right click and I can choose copy analysis to group. This is the condition that you need to have in order to create a template that works. Because if there's no gating strategy associated to it, FlowJo will not know what to do. It knows that there is a group, but it doesn't know what to do with that group. And whenever you create a template, this will be an empty workspace. So in order to make it work, you will need to have a gating strategy that is group owned. And then at that point, you can save it as a template. Okay, so the group needs to be live and the gating strategy needs to be group owned. So then if you want to save your template, you just go on the FlowJo icon up here and you select Save As Template. You will realize that this template will have a T final. It's not WSP anymore, but it's WSPT, which means template. So I can save this template to the desktop. And then if I open it, you see that it's empty, but there is a gating strategy there. So if I now add some samples with the informations that I inserted before, which are like including LD in their name, for example, if I load these samples, they are automatically loaded into my group with those conditions. And if I look down, I see that all my samples are actually analyzed with the same gating strategy. So if I open up my plots right here, I can then navigate to the next sample. And you see that this gate is slightly not completely centered, let's say. So I would either move this gate. And in this case, since my group is not synchronized, I'm only changing this gate. So if I'm going to my next sample, it's still in the original position. If I'm going on to the next sample, it's in the original position, and so on. So if you make your groups synchronized, so I can right click on my group, edit group properties, and make these groups synchronized. If I apply the changes and close, next time that I move a gate in any of my samples, this gate will be automatically moved to the rest of my samples. So depending on the kind of analysis that you need to do, if you need to keep fixed gates or gates that are exactly the same within all of your samples, you can make these groups synchronized. Otherwise, my suggestion is not to have them synchronized, because it's really frequent that you have shifts of populations from like slightly small shifts of populations that you can have from one sample to another. So in this case, I wouldn't make it live. I wouldn't make it synchronized, because you may need to move your gate a little bit. One option that could actually help in automatically move the gate into the right position, which is something different. So it's not a fixed gate, it's not an individual gate, it's a gate that would automatically move if the population shifts. Something that you can do is making this gate, going to active gate, and make this gate magnetic. This will bring the gate to automatically move, okay, to the more dense population. So in that case, I can place my gate here, and it will automatically move to the closest and more dense population. If I apply this gate to the group, now it should automatically move to my population, as you can see here. And you see the arrow saying the distance, the size of the arrow indicates how far was the gate position. That's pretty much good. In this case, you see that the gate was a little bit shifted, and so on. And then whenever I also create a template, I can create as well a layout editor setup. So I can set my layout editor to get exactly the same gating strategy or the same layout that I want to have. Let's say, for example, we want to look at our gate right here, and we want to have an ancestry next to it. This ancestry can be vertical or horizontal. So let's say I want to have it horizontal right here. I want to add some informations, for example, regarding my sample. I can create a text box. And whenever you're using automatic annotations, that can be, for example, if you right-click on the text box and you want to insert annotations such as the FlowJo version and the date, these are automatically inserted by default on the date that you are working on. So if you use these features like this one, whenever you save it as a template, next time that you load your samples, this will be automatically analyzed, and it will also create automatically your layout. And in case you didn't know, if you turn off, if you move from iteration off to iterate by sample, you can actually navigate through all the different samples and see exactly what FlowJo has done on your samples, so you can quickly scroll down to all of your samples. So what are actually the conditions to have a template that it's actually working well and is functioning? For sure, having the same panel and staining panel, and having a group and a gate and a group-owned gating strategy. The group must be live. especially if you select some characteristics for the file name samples, for example, to be included in this group. And that's pretty much it. So why do you need to, why the template will work on the same, on samples that have the same panel? Because whenever you create a gate, FlowJo applies those parameters to the gate, right? So if you, let's say, stain your samples with CD45 FITC one time, and then you want to use that template on samples that were stained with CD3 FITC. So these are different staining names or different parameters eventually. FlowJo will just use a combination of those parameters. And if these are not matching your new sample, then this is going to be difficult for the software to find the exact match. So you may see like knot populations show up. So these are the conditions to have a template actually working well. So group owned gating strategy, eventually make the group live, same panel, same staining. And that's pretty much it to have all the templates working perfectly. So let's see if in the meantime we got any question in the Q&A. No? Okay. You can also ask me, how am I doing today? I don't know. Okay. Okay, so let's see if I have another question. Oops. Okay. Okay, so looks like another question that came through is okay. What is that BD icon on top of FlowJo left screen? So that would be this one, okay? So if you ever wondered what is the BD icon on the top left corner of FlowJo, this is actually a connection to one of our platforms, okay? So as you may know, FlowJo joined BD several years ago. Actually, when I joined, oh, we had another person online. And so you may have noticed that there is this connection right here to something from BD. Of course, we are part of the same company, but what is that? So if I click on that, this will bring me to a new platform that is called BD Research Cloud. BD Research Cloud is a collaboration environment where we can actually make or do many things, including designing panels or store our data and so on. In order to access BD Research Cloud, you would need a portal ID, a FlowJo portal account, which is basically the same thing that you use to log in into FlowJo if you are using a user subscription, and it's a free account. So anyone can create one, even if you don't have a FlowJo portal account for the FlowJo software specifically, you can still create one. You can just go on the BD Research Cloud, or you can just type it, or you can just access through FlowJo, and you will be prompted to this window right here, and you can just create an account if you don't have one. Again, it's free, so in case you're interested, just give it a try. Now I have my portal account, which is actually the same that I use in FlowJo, and I can sign in right here. And there are many things I can do in this area, starting from designing panels to create Diva experiments, to set up workflows for my lab or for my panel design. I can load and store my data. So no need anymore of going around with a USB stick. Or if you just want to share a workspace with your colleagues, don't need to save it and save the FCS files and so on, and then send them and then use an external thing to drop it and so on. So you can share absolutely informations with your colleagues. This could be either panels or data or workflows or experiments and so on. And then you will have some extra features right here, such as loading cytometers that you are working with. The reason why, or the action that you can perform when you load cytometers, it's telling the platform which cytometer is available for your lab or for your experiment. And based on this, based on the configuration of the instrument, BDRC will know whenever you are designing a panel, which are the fluorochromes that can be read on each of these instruments. And so it will help you during your panel design to propose you, let's say, just the fluorochromes that can actually be seen on that specific fluorochrome. So this is on that specific instrument. So this is already a help in case you want to design panels. You will have also a list of reagents, and you can create custom reagents in case you need, and then you will have informations regarding most common populations that are out there and how to characterize them. Let's say, for example, here I have a list of literature-defined populations. So, for example, if I click just on the CD8 factory memory T cells, I can see if they are positive or negative for specific markers and which are the markers that are suggested to be used in order to characterize these cells. And of course, you can find a citation link to the paper where these informations were taken off. If you have your custom population, because that's the best thing, right? Creating your own population, characterizing a new population is mostly our goal. You can create your own right here. So you can just click create and have your custom population. What this population will do later on, whenever you design a panel, okay, you go onto the panel section, and you will be prompted with published panels. So you can find all the different panels that are published. The latest one is the OMIP panel with 50 colors. So that's really a lot of informations that you can include in a panel. But we also have other different OMIP panels, which are actually published panels. And as well, you will find some BD defined and validated panels. So, for example, a 33 colors panels, 3 laser panel to characterize the B cell subset or the T cells senescent exhaustion, and so on. And then you can also find specific backbones panels that you can then combine. These are called panel blocks. And if you load more, you will be prompted with more and more panels that you can eventually combine. In this case here, these are all backbone panels that can be combined in a single experiment eventually. And you will also have as well the ability to drop in some extra markers in case you're interested in that, okay? So you can create your custom panel if you are interested, and you will just need to go on to create panel right here, assign a name to the panel, assign a project, and then start. So this is just like to design panels. For example, you will select the instrument that you will be using and the configuration, and then fine, I can do it with a FAC Symphony A1 or a A5 SE or S8. These are the ones that I have in my lab, let's say, or at home just on my desk. Okay, so I can complete the step, and then I can go on. And then you can select either a non-specific species, let's say you're working with plants, for example, or you can select a human, mouse, baboon, and so on. And then you can start either adding markers or populations. So you can add, for example, T cells. I can add my naive CD8 T cells. Eventually, I can look for my B cells. And while you are adding more populations, these can also be your custom population, or you can add markers individually. Okay, let's say HLA-DR, for example, add. While you add more markers here, you will find the species, and then you can define if it's an exclusion marker or not, and you can also assign row densities and say, okay, this is really highly expressed, this is like mid or low expressed. And you can define, if you have defined populations, it will tell you already this is positive or this is negative, this we don't know yet, and so on. So once you are happy with your markers or to define or using markers that you need, you can just complete the step and go on. And then you can start assigning the fluorochromes to your markers. So based on the configuration of the instrument that we gave, BDRC will prompt us with several fluorochromes that can be associated to each of these markers right here. And on the left side, you will see the spectrum and each laser that you have. So I can just select the first one, which would be my CD3. Now, I'm not an expert in panel design, and I'm an expert in FlowJo, so I will just assign this randomly, and please forgive me if I make any mistake. But just to say that here you will have the fluorochrome name, the primary lasers, the peak filters, and the brightness of the fluorochrome. So just thinking of the basic rules for panel designs, you want to assign not so bright fluorochromes to markers that are highly expressed, and eventually bright fluorochromes to markers that are not highly expressed. So eventually I can use like a mid one, this one, and you start pairing, and you see that the spectrum starts to fill in. So I can pick, I don't know, this one, okay. And then whenever you have something already occupied or something problematic, BDRC will either gray that out or will highlight that with a red square around it that says, look, this could be problematic because BV421 uses the same peak filter as B450, so it's not a good idea to combine them. So maybe I can go on to a different one and so on. And then you keep going on. And by the time that you finish, and again, just randomly assigning them. I have tons of things available right here. I can look at them individually, or I can stack them. So I can see for each laser, violet, blue, yellow, or red, what do I have on those lasers? And then I can just complete the step. So whenever I complete my step, it will suggest me eventually to add a brilliant buffer because I used brilliant reagents. And then I can as well complete this step. And then right here, I have the options to finalize my workflow or to save this step and come back later. You will have the informations on all the different antigens that you are using, all the different fluorochromes. And if I finalize this workflow, I can then eventually, for example, send these as an email to someone, or I can download it, or I can view my panel and say, okay, nice, this panel was done on a FACS Symphony M1. These are all the reagents. I can visualize how it looks like. I can add a staining sheet to that, and I can share it. One more thing that you can actually do with that BD icon on FlowJo is to load data. So if I open up my FlowJo now, and I want to add some samples, I have the options to add samples from BD Research Cloud. So what that will do will automatically connect me with my storage area of BD Research Cloud, which is basically this one, where I have all my data, and I have different projects, okay, that I share with people, and I also share my data with them eventually. So for example, let's say BDRC demo, I have some project files here, and these are all my files that I can automatically load into FlowJo, or eventually I can share or I can download. And one thing that I really find useful whenever you want to save a workspace and share it with someone, it's just to go onto the FlowJo option right here, and then you can set it as save as BD Research Cloud workspace. So at that point, you will have a workspace in BD Research Cloud, and then you can just go in there and share it with your colleagues. So that's what the BDRC tool does, or the BD icon tool does within FlowJo. And these are the general functions, okay, the ones that are most connected to FlowJo itself. But of course, you can generate Diva experiments if you are working with Diva. You can generate workflows. These can also be not only panel design workflow, but also instrument workflows. Let's say I need someone to start up the instrument, shut down the instrument, and so on. We have seen data, we have seen cytometers. In reagents, you can also generate your inventory. So let's say you have some fluorochromes in the fridge and you want to create an inventory. You can go onto my inventory, and if you are like me now with a basic license, you can go here and activate your free trial, so you can try also the inventory one, which will allow you also to scan the reagents with your phone. So once you scan the reagent, you can just look at the reagent that was scanned and look at all the informations, who did that, when they did that, which is the catalog number, and so on. And in the reagents area as well, you can create your custom reagents. We don't and can't have all the reagents in the entire world, but if you do have one, either you scan it or either you create a new reagent on the top right corner. And finally, you can use the spectrum viewer if you are not designing a panel yet, but you just want to know, for example, if two fluorochromes are doing well with each other or not. You can just select the cytometer here, define which cytometer do you have, which is the configuration of the cytometer, and here you will have the filters that you have, and then you can just select any of the fluorochromes. So let's say that you do have some fluorochromes in-house, you have some markers already with some fluorochromes attached, so you can just go in here, pick whatever you have. Let's say I have CD10 in PE. Okay, I can see if it's the correct one, and then I can add it. And so I can see the emission spectrum of CD10PE. Then I can add, I don't know, something else, FITSI, okay, light chain. And then instead of going here, I can do this and see if I have overlap within my fluorochromes and so on. So this is more about panel design, but if you're interested in panel design, there are many, many resources on the BD website. And here in groups and projects, I can generate my groups and projects. And indeed, you see, I have a project by myself that no one can get in. I have some informations in here, which are the users. It's just me, which are the workflows and the details of that. But I also do have some shared projects with someone else where I see shared groups where I see the users and I see the project in each of these group. Okay, so I think that's quite a fair explanation of BDRC, but if you do have more questions or if you are interested in knowing more about BDRC, there are webinars available on our webpage. So if you are here, it means that you either were on our webpage or you saw this on LinkedIn. So if you go under the Learn area, webinars, this was done yesterday, so it will come up under the recorded webinars anytime soon. If you're interested in any upcoming webinar, you can sign up here. This will be about FACSDiscover assay data analysis, but if you are like more on a, let's say, starting point with FlowJo, you can go on all the recorded webinars and find, for example, a specific webinar on compensation. This was done only on compensation, starting from the theory and then going on to all the practice and all the different examples of compensation that you can have in FlowJo. Or, for example, introduction to FlowJo. These are basic webinars, or high dimensional analysis webinars. So if you're interested, depending on what you are doing, you may know that with FlowJo, you can do many things. You can do basic analysis, you can do advanced analysis, you can use algorithms and machine learning and so on. So it really depends on what you are interested in. There is one on spectral data, there is one on QC and normalization, another Q&A live. So maybe your question was already asked in the previous Q&A, and so on. So just in case, remember that all the webinars are recorded and you can access to that. Eventually, remember that also you can access the FlowJo University. FlowJo University is a repository of short videos on everything we have. So we have the FlowJo area, SeatGeek or BD Research Cloud area as well. So if you want to get started with BD Research Cloud, this is also something that you can watch. Or if you're interested in FlowJo and you have like some specific questions, I strongly recommend to give a check if you are an expert user of tips and tricks in FlowJo, for example. There are many tips and tricks that you can have, like many times we cannot file our files in FlowJo. How do we reconnect them? Or what is an archive file? Changing the stain names if I made it wrong, and so on. How do I add keywords and so on? So I really like the FlowJo University because it really gets you the answer right away with a video, and it's really, really useful. And we have that like for the start, for the naive, let's say, for people that are starting using FlowJo. So you can just go on getting started in FlowJo, how to install it, what is the workspace, what about preferences, and so on. So let's say no Q&As still, okay. So then I will probably go on to my next question that I received. So the question is, when I have multiple variables, do I think to make multiple concatenations? Okay, so what does it mean having multiple variables? It means that you want to compare samples that are coming from, for example, from different conditions, and you have multiple donors, okay? So one variable will be donor, the other one will be condition. So in this case, you have two variables, and you want to compare them, and you want to compare them by using high-dimensional analysis. Now, I don't know if you are familiar with it, but in the latest, let's say, couple of years, people are analyzing their data by using algorithms rather than manual gating analysis or manual gating strategy. And honestly, I'm really happy with that because myself, I'm so tired of making manual gates. So I really appreciate that. But in order to use these kind of tools, you need to create a single FCS file from whatever you want to compare, right? So if we want to compare multiple samples from multiple treatments, we will need to get a single FCS file out of this. And this is needed and stems from the nature of the algorithm itself, because these algorithms are stochastic. So we need to work with a single FCS file. Doesn't matter which is the platform. If it's FlowJo or it's another platform, everyone will need to work with a single FCS file if they want to move on to high-dimensional analysis, which could be, for example, a t-SNE plot, a UMA plot, and so on. So this action of creating a single FCS file, it's called concatenation in FlowJo. So now the question is, if I have two variables, do I need to do two concatenations? First concatenate my donors and then concatenate my treatments? No. The short answer to that is no. You need to do only one concatenation step. Okay. And what you want to do before concatenating is assigning keywords to your data. Keywords are metadata, so are extra informations that can be added to your files. We are not changing the name of the file, we are adding extra information to the file, and are really useful and are needed, especially when you do high dimensional analysis. So let's say that I'm gonna add now a couple of samples, okay? I have them here. So I have two patients, two conditions of stimulation, okay? So the question is, do I first concatenate my stim and then my non-stimulated into different files and then I put everything together? No. What you want to do is add the keyword, okay? So how do I add the keyword? I go onto my workspace right here, or under FlowJo or under workspace. Workspace will show you all the keyword tab with all the different functions. FlowJo will only show you the add keyword tab. So first I need to add a keyword, a variable, which is, for example, donor or patient. Okay, here I have patient 1 and patient 2, non-stimulated, patient 1 and patient 2, stimulated. So I will add one keyword for my patients. This will be patient 1, oops, and patient 2. This will be 1 and 2. So whenever you add keywords for your high dimensional data analysis, it must be numeric keywords, okay, not words, because when we plot them on a bidimensional plot, we don't have way to scale with words, but we scale with numbers. So the first one will be patient. The second one will be treatment or condition. Call it the way you want. Doesn't really matter. So I can define the non-stimulated one as my condition 1. So this will be condition 1, and this will be condition 1 as well. And my stimulated ones as condition 2. So this will be condition 2 and condition 2 as well. Okay. So then when I go into my concatenation step, of course, you want to clean up first as much as you can, please. And I'm really a freak with cleanup. So if you ever end up to watching my QC and normalization webinar, I'm really focused on cleanup. So you want to clean up these files. You can use algorithms for that, or you can use manual gating. Okay, so let's clean up at least here. And then at least exclude the dead cells, sorry, the doublets. Okay, let's at least do these. And then I think I have a viable marker here. Yes, here it is. So at least clean up on physical parameters, exclude doublets, debris, and dead cells. These are my live cells, okay. Then I can either move these ones to my next samples. And then I want to concatenate. So now that I have two variables here, what I can do, I can select the populations that I want to concatenate, and I can right click on that and choose select equivalent nodes. This is let's say a tip that I give you to make it faster. So this will select exactly the same population at the same level with the same name. And then I can go under files, export concatenate populations. Okay. At this point, you have two options. One is to export the populations. The second one is to concatenate, and I want to concatenate. Please make sure you have a destination folder which is real on your desktop, because FlowJo keeps in memory the last one that you used. So if I go onto my desktop, I should have, I don't know. No, I don't have one yet. Okay, so I will just do it on my desktop right here. And then another advice that I would give, okay, you can choose to take it or not, but I prefer to compare similar events or the same number of events within the different conditions. And this is like to give a sort of normalization within my samples, because Otherwise, whenever I compare 1 million of cells versus 10,000 cells, wouldn't make that much sense, right? So many people perform downsampling on that. There is a shortest way if you're willing to do that. And instead of performing the step of downsampling, when you concatenate, you can actually choose include no more than. And this is exactly the same thing of downsampling, and tell how many cells you want, how many random events you want to be picked up from each of these populations. So I can say, for example, 10,000, and I can then go on to my advanced options. And since we are here, another frequent question that I get is, should I concatenate all compensated or all uncompensated parameters? Now, the answer to this is, let's say, quite easy. So if you are concatenating files that have the same compensation matrix, like in this case, as you see here, I have the same compensation matrix for each of my files, I can choose the all uncompensated parameters. And when I will create my concatenated file, I will find again my comp file, my comp parameters that FlowJo made for me. If I do have different compensations on my samples, then I want to choose all compensated parameters, because if I move with the all uncompensated parameters, what happens is that FlowJo, among the different compensation matrices that are there, doesn't know which one to pick. So, and to assign to the new file. So you will have a file with no compensation matrix attached. So in that case, select all compensated parameters. FlowJo will make, let's say, a unique compensation matrix for all of them, and you will get a file with a compensation matrix. Now, we are almost done. We can assign a name. Okay, and we can choose to concatenate all the files together that we have chosen, or just every n files together, and then files, for example, with equal keywords values. But in this case, we want to concatenate everything together. And then we don't have to forget, when we want to create such a concatenated file, to choose additional parameters, which will be our keywords that can discriminate individual samples and individual treatments. So in that case, I can go on to additional parameters, choose, and then select, what was that? Condition, I think, patient and condition. Okay. Now, and I got also this question in the mailbox. In the concatenation dialog, there is a checkbox that says spread distribution of keyword data. What does that do, and when should I leave it on or off? If you hover over, you see that it gives you an explanation of what it does, adds some random noise to the value of keywords so that they form clusters around the actual value. Well, you don't want to have that. You want to have the actual value without any random noise next to it. So you don't need to spread the distribution of keyword data. And then you can click concatenate, and then eventually you can open the file onto a new workspace or onto the existing one. So I can choose to open it onto the existing one. And now I have a compensation matrix attached to it, because all of them have the same compensation matrix. If I double click on this file, I will see the full concatenated file. But if I scroll down my parameters, I will see also that I have new parameters here. The sample ID parameter will be created every time you generate a new concatenated file. So you can imagine that if you make a double concatenation, you will have two sample IDs, which will make you crazy to understand what you are doing. If I plot it, you see this is sample 1, 2, 3, and 4, and they are ordered from left to right, as on my workspace from top down. But I don't want to use this. I want to use my condition 1 and 2, and eventually I want to use my patient. Oops. Okay, let's do this. Instagram. Yeah, here it is. So I can either gate on my patients or gate on my conditions, or I can look at them at the same time. So in this case, I can just gate, this will be patient 1, and this will be patient 2. Okay. Or I can do the same thing with condition. This will be my condition 1, so non-stim. And this will be my stimulated. Just this. And so then you can proceed with your analysis, you can do a t-SNE and so on, and then you can backgate each individual condition or patient to that t-SNE plot, UMAP plot, and so on. So that's how you actually behave, or how you go on whenever you want to compare different conditions, different treatments, and so on. You will need to use a single FCS file, and keywords will be the thing that will help you to combine all these things together and to assign each individual characterization to that, let's say. So if I use keywords, then I can know which one is patient 1, which one is patient 2, which one is condition 1, and which one is condition 2. Okay, we are at the top of the hour.